So good afternoon, everybody. Welcome to this seminar, the Xylella Seminar. And I have the pleasure to introduce Valerio Mazzoni. Most of you know him. Valerio is from the Fondazione Edmund Mach in San Michele Adige in Trento, in Italy. And he will talk to us about biotremology and the approach to interfere with the Xylella vector. So I let the floor directly to Valerio for his presentation, and thank you very much for joining us today to present about this interesting topic.
Thank you, Giuseppe. So thank you for the invitation. I'm very glad to be here to talk about this topic. I start immediately to share my screen. And please confirm that you can see my screen.
Yes.
Okay. So as Giuseppe said, the title of my presentation is about the approaches that biotremology can use to deal with the Xylella fastidiosa, in particular behavioral manipulation towards vectors. This is the work that I'm summarizing now that started more or less 10 years ago, 2014, in the US. And I'm going to talk about two main experiences I've been working with since 2014. On the left, you see Omaldisca vitripennis. It is the glassy wing sharpshooter. And on the right is Spittlebug Philenus spumarius. Both of them are known as vectors of Xylella in the US on the left side, and in Europe on the right side. So, but first of all, a short introduction about biotremology. And I must say that this is a discipline relatively new. The first definition was done in 2016. You see on the left the primer published by Peggy Hill and Andreas Wessel, and the title was Biotremology. And this was the first time this term was used in the way we know today. And more recently, in 2024, so this year, we came out also with an official definition of biotremology, which is the scientific discipline that studies interactions between organisms, which are mediated by substrate-borne mechanical waves, which propagate along the boundary between two media. And in fact, as you can see from the figure, the red line on which this plant hopper, the spider, are staying is a boundary between air and the substrate in this case. So this is the definition that is accepted, and biotremology, however, after less than 10 years of existence, is already spreading, and there are books dedicated on the Springer editor, but also Facebook group, YouTube channel. There is an official logo, and this year our community organized the 4th international meeting in Canada, in Toronto. So the movement is going on. This is just to remind everybody that this is a common phenomenon. It's not something obscure or rare, because we have a bias about acoustics. In fact, if you consider how many groups use acoustic communication, so this means airborne communication, well, you can see from these dots that it's not really very spread in the insects. But if we consider vibrational communication, we have more than 200,000 species that are known to do that. So basically all insects use vibrational communication. Therefore, this is a phenomenon that is widespread, and for this reason, worthy to be better investigated and known. And also the terminology had an important update this year with a paper that we published on plant science, where we introduced the terms of pherodons and allelodons, where this term in parallel, of course, with the pheromones, allelomons, it comes from the Greek word doneo, which means to shake. So basically we have pherodons that mediate intraspecific interactions, allelodons interspecific interactions, and of course, they are then divided in chirodons, allodons, and cyanidons. So we have a perfect parallelism with the chemical ecology. In this talk, I'm gonna talk about two methods of behavioral manipulation. And this example you see on the left, vibrational emitting disruption of VMD, which is currently largely investigated, and also experimental fields occur already in several countries in Europe to control Scaphoideus titanus, which is a vector of flavescens dorsale and phytoplasma. On the right, the other method of vibrational behavioral manipulation is the vibrational attraction, where the vibration acts as a lure to make traps or other strategies like attract and kill. In this case, we've been working with Allomorphalis for many years, and now there is a commercial trap available for the market and for the farmers. So basically, when we do behavioral manipulation, we need to respect some prerequisites. As you can see from this table that I made, to do vibrational emitting disruption, it's very important that we have disruptive or masking signals, of course, that the insect use mostly, I would say clearly, one channel of communication, which is, of course, vibrational communication, so vibrational signals. And they use these signals for identification and search of the partner. And it would be ideal to have monophagy in the species. As for the lure approach, instead, it's very important to have attractive signals. Again, the insect must use these signals for identification and searching. And it's very important also that the insect that lead the duet, we will see later what this means, is not the searcher. So this means that because there are specific rules that we will see that must be respected when there is an ongoing duet of vibrational animals. And in this point, it's important for approaching with the allure strategy. So let's talk about Malodisca vitripennis. And this is a sharpshooter, and so it's a silent feeder, of course. And it is nowadays spread in Southern California. At least this is the area where I was doing my study. And you can see that the green area is also the agricultural part of California, the Central Valley mostly. And you see that this area is covered by almonds and citrus and vineyards, of course. And in the Bakersfield area, which is the Kern County, we have a situation with an important impact of Pilz disease that was calculated in 2014 as more than $1 million per year, but nowadays this estimation is raised to almost 200. So this means that there is an important impact of Xylella, and with strategies that everybody knows, with the use of insecticides and removal of infected plants. What we did in the period between 2014 and 2017 was a classic strategy to approach the behavioral manipulation. We start with the mating behavior, the study of mating behavior vibrational signals of the target species. Then we do playback tests in the lab to assess the role of the signals. Then we do still in the lab mate interference trials. And finally, we go into the field to test them. We stopped at this level in this project, and however, it would be important to go further to higher level or TRL in order to make these devices available for farming. So what we did, this is the first step, of course, the study of the mating behavior and the pheromones, so the vibrational signal intraspecific associated to the mating. And you see that the behavior, this is a Markovian chain, and an ethogram. it's pretty complicated. There are several signals, 3 male signals and 2 female signals that, of course, are used by the insect in a duet way. So this means that the male and female alternate their signals, and there are specific paths that must be followed to reach the mating. But therefore, it's a stereotype behavior, but with precise rules and the specific use of vibrations to achieve the mating. However, we found also male rivalry signals. This means that males can rival one another to reach the female. And in fact, the title of the paper that we published talk about mimicry. This means, and you can see from this graph on the right side, that the male signal or rivalry are not distinguishable from the female signals. This is because the insect, once is aware of the presence of another male, try to attract it, and so there is a natural distraction to prevent the other male to reach for the female. Then once they are close, the male that attracted the other one start a fight to let the other be removed by the plant. Of course, this is a strategy using a disruptive signal, which is working pretty well. So this strategy is important because these are prerequisites that we were talking about before. All the first 4 prerequisites are respected in the case of Omalodisca vitripennis. Unfortunately, this one is not respected. So this means that the searcher, which is the male, is not the one, is leading the duet. This means that it's difficult to make a lure for this because it's pretty complicated. Omalodisca is not monophagous, so there are many plants that can be host for the species. However, in this case, it's not a big problem. What we did was to test the vibrational mating disruption against these species. And so basically, we started with laboratory experiments, and we tested 3 types of candidate noise. The first one is called female signal 1 noise. So there is a series of female signals. The second one is a natural female signals. And the third one is a white noise. So it's a noise whose frequency has the same energy in all the spectrum. And of course, the silence as control. What we see here on the left, we have in general 20% of mating. So this species is pretty lazy, I would say. When we put together insects, only 20% without any disturbance in half an hour. That is the time we gave them to achieve the mating. However, we see that the female signal was disruptive because we were basically using a signal in the way a rival male does. And the white noise also create a masking effect, so there is no way the insect can perceive the presence of the others. However, even the female noise, so the series without any break of female signal was perfect to compromise the mating process. And here it's interesting on the right side, once the playback was stopped, there were matings, even at higher level, although not statistically significant, but this means that the insect immediately after stopping the signal of disruption were immediately able to start a communication in mate. This is another playback experiment that we did, and you see we have 5 types of female signals. The first one is the natural signal that we use as control. Then we have number 1, 2, 3, 4. What are the differences? Well, in the case of number 1 and 2, we simply made the lowest part of the frequency. These are spectrograms, okay? Just to be sure that you understand. So basically, we go from 0 to 1000 hertz, and there are many harmonics, which is a classic female signal. We tried in FS1 to make the lowest harmonics lighter. So this means that there was a big prevalence of the highest harmonics in the signal. And in FS1, we deleted them. So there were only higher signals, harmonics, sorry. In FS1 and 3 and 4, we did the opposite. We made the highest harmonics lighter in FS1, number 3, while number 4, we deleted them. So basically, the idea was to play back this signal to males, and see how many males were responding to these signals. This was important to understand what are the structure of the signal that really are attractive, make the signal identifiable to the male. And you can see from the right graph, we have interesting results with the number 2 and the number 4. That is when we deleted parts of the female signal, and there was a big reduction, a significant reduction on male responsiveness when stimulated with these two signals. So this means that if we prevent the male to hear, to perceive a part of the signal, so not the full signal, but it's enough to prevent part of the signal to be received by the male, we can compromise the reception. So what we did, that is described in this paper, that the aim was to design a candidate vibrational signal for mating disruption. But what we did, we created 3 different disturbance signals: number 1, number 2, number 3. Number 1 was covering 80 Hz, so the lowest part of the harmonics. Number 2, 240 Hz, so the higher part, so middle high, I would say. And number 3, both of them. And here we had very interesting results, because when we use a normal signal, all males responded. But when we used together with the signal, one of these disturbance noise signals, we had a total disruption in the case of number 1 and number 3, a bit less in number 2. I mean, it was still significant, but still 30% could respond. But in the case of the number 1 and number 3, so when we were masking the 80 Hz, so the lowest part, or of course, even better, both, there was a total interruption. So basically, we created a disturbance signal, and we went to the field at this point, together with the colleague from USDA, Rodrigo Krugner, Parlier. We are in Central Valley, near Fresno, and we made an experiment with 134 individuals that were released inside the nets on the plants. And of course, we used disruptive signals that were transmitted to the plants thanks to these shakers that were attached to the wires in this case. And you can see from the pictures that this was the trellis that were typical of that area. These pictures are taken later in the season, of course, but just to show you the trellis. And results, and this is the nets. These are the nets where we were releasing the insects, couples, and stay there for 24 hours. And we, of course, in the control, there was no disturbance. Why in the test, in the treatment, we were using a disturbance noise. So basically, these were the results. We had 21% of mating in the control, which is consistent with our trials that we did in the lab, where we had this 20%. But when we use the disturbance noise, that is the designed disturbance noise that prevented in the lab the disruptions, also in the field, 0.6% of the individuals made. This means that only one pair out of 134. You can understand that this is a very interesting result. And however, the research stopped because fundings were finished in that moment. And however, we think that it's really promising to use this technique against these species. And now for the second part of the talk, I'm going to speak about Philenops spumarius and our experience in Italy. You know that the problem of Xylella is widespread, and the pictures from this side of the slide regard Apulia in southern Italy, where we have the problem with Holy Creek decline syndrome. And the problem is very similar to what we observed in California. With in that case, it was vineyards, and here these are olives, but the perspective seems very similar, and also the way it is managed through the large use of insecticides and the removal of infected plants. In this case, the investigation started in 2020, but is still ongoing. At this point, also including a period of lockdown during the coronavirus crisis, however, we could accomplish 3 steps of the experimental progress, and we described the mating behavior of and associated vibrational signals of Philenops spumarius. We made the playback test to assess the role of the signals, and also we did tests in the lab that involved not only mating, but in this case also the feeding and the role of the vibrations emitted by predators and other insects that can occur on the plant, just to extend our playback setup and see what are other kind of vibrations, what are the consequences of other kind of vibrations on the behavior of Philenops spumarius. So in this case, these are some papers that we published, and what we observed is that, of course, substable vibrations are fundamental for intraspecific communications, and this was not surprising for us, but it was important to describe them, of course, in detail. And what we asked ourselves when we started was, but intraspecific vibrations can be, and also we will see later also interspecific vibrations, so pherodons and allelodons, can be used to disrupt vector plant bacterium interaction. So in this case, we also investigated not only the mating, which is something we did, but also a possible interaction with the bacterium. And in particular, at this stage, we are still investigating the possible disruption of feeding. But our research will go soon to investigate about the ability of acquisition and then transmission. This is the behavior of Philenops spumarius. Consider 9 periods, starting from late May, when adults are in the field, in the condition of southern Italy, to period number 9 and 10, that is the end of September, October. So basically, what we can see from these graphs is that the male signals, MCS, are present throughout the season. However, female signals appear only from August, I would say mostly, and the duet between male and females are very late in the season. So basically late August and September, when also we have matings. So the species is in the field for many weeks, but until the end of the summer, there is not mating behavior. And this is also something we investigated at the level of physiology, and in particular, egg and ovariole physiology. What we see from this graph in pink is that the females have not ovarioles ready, and so eggs ready, until the end of July, early August. And this is consistent with the behavioral observations and the emission of signals that we observed in our trials. In fact, you see from this graph that calling females and females with ovarioles are really in parallel during the period of study. So what we observed in this case, again, a situation extremely similar to what we observed in the US with the glasswing sharpshooter. And again, we consider more promising the use of vibrational mating disruption against Philanus spumarius rather than an approach of attract for trapping or attract and kill. What we did then at that point was again going into the lab and making tests. We tested in this paper, we tested two different type of signals. In test 3A, for instance, is a female rejection signal, because the female can reject the male, especially in the first part of the season when they are not available. This B is a male rivalry signals, basically, and then this is a noise that we introduced as another variable to be tested. And however, what we see, there is no significant differences in the number of mating or, for instance, a number of duets, number of copulation between the different signals and the control. This means that playing back these signals did not affect the behavior of the insect. However, when we tested this noise number 2, which is covering exactly the frequency of the harmonics of the signal of Philanus spumarius, and we can see in these other graphics. So we created this noise 2 specifically like we did in the case of glasswing sharpshooter, with the aim to cover exactly the harmonics. And in this case, you see the number of duets and number of copulations was dramatically reduced. So basically, we found the signal promising, very promising for mating disruption. Another aspect that we were investigating is about feeding disruption instead. In this figure, you can see that we tested with the technique of EPG, electrokinetography, the capability of the insect to feed from xylem when disturbed with the different type of playback signal. In this paper, we tested this one cis15 and cis50 that were two types of interspecific signal that we tested. And you can see that the cis50, 15 is an amplitude value, so it means that 50 was much louder than 15. However, when we disrupted with the cis50, there was a reduction. In the higher graph is the total duration of silent ingestion in 3 hours of recording, and in the second graph, the percentage of the probing time spent in silent ingestion. So compared to the control, there was a very important reduction of these values. And then we did other experiments about the possible use of other natural signals, in either inter or intraspecific. In this case, we assessed two important behavioral aspects, motility and then probing and feeding again. And so basically we tested two types of spiders, that is Xysticus cristatus, Pisaura mirabilis, that are known to be predators of Philanus. We tested also the ant, Crematogartes pudellaris, and also Staphylococcus titanus just as a sort of control of an insect that shouldn't be a problem for Philenus spumarius. Of course, we tested also in other intraspecific signals and general noises. In this case, what we found for the motility, a clear freezing behavior every time we play back the spider Cystico cristatus. This means that the insect stopped any activity and didn't do anything as long as the signal was on. As for probing and feeding instead, we observed that when together with the spider, also the sound of the vibrations produced by the ant Crematogastes scutellaris caused a high level of escaping from the plant, so a sort of signal that can cause repellence on the plant. Other aspects about probing and feeding, again, you see on the left we have the control. Crematogastes scutellaris caused an important reduction in terms of both xylem contact and xylem ingestion. So basically, I come to the conclusions. I think I'm at the end of my half an hour of talk. And I want to say that both species, Omalodisca vitripennis and Philenus spumarius, use vibrational signals as main and I would say almost exclusive communication channel for mating communication in first instance, but also for other aspects of their life, and that both mating and feeding, there is a mistake, mating and feeding are susceptible of interference when using proper and specifically designed playdecks. In fact, we could observe and demonstrate with the laboratory and in the case of Omalodisca vitripennis field trials that the use of disruptive vibrations can compromise significantly mating, and in the case of Philenus, we also observed an important disruption of the nutrition from the xylem. And yes, of course, we expect in the prosecution of our research to go more into the fields and trying to work with these principles in commercial vineyards or orchards in general. So, this is my last slide. I want to thank all these people that were really crucial, important to do the mass of work that I quickly showed in my presentation. And I'm fully available to respond to curiosity or questions that you can make to me. Thank you.
So thank you very much, Valerio. This for a very, very clear and very interesting talk. There is already one question in the chat.